Good day, this is Professor Will, CBMD PhD. It's Thursday, November 21st, 2019. It's 4:39, 16 seconds Eastern Standard Time. And today I'm going to be doing a reading on microorganisms. So let's get started here. The organisms inhabiting the soil span a wide range of taxonomic functional groups. As many as 1,000 to 5,000 different genotypes may be present in a gram of soil, based on the re-annealing kinetics of DNA extracted directly from soil and then denatured. Archaea and Eukarya, domains that are differentiated by sequence patterns of small subunits of ribosomal RNA, bacteria and fungi are the most abundant groups in soil with respect to biomass and numbers. Archaea, including methanogens, extreme halophiles, and extreme thermophiles, are present, but their significance in soils are neither flooded nor exposed to environmental extremes is not well understood. Phototrophic bacteria and green algae are abundant only when soils are flooded. In wetlands, rice paddies, poorly drained soil. Viruses and bacteria, fungi, fauna, and plants are common and exist both in and outside of their soil-dwelling hosts. Many organisms in soil are not metabolically active but appear to be in wait for an opportunity when environmental conditions favor them. Soil microorganisms are also categorized on the basis of the functions, usually metabolic, they perform. All life forms require a source of electrons, electron donor, an electron sink, and a source of carbon. Metabolic requirements in these three categories form the basis for a useful functional classification system that targets processes more than individual organisms. Organisms are classified as phototrophs, lithotrophs, or organotrophs, according to whether they obtain energy from light, inorganic chemicals, or organic chemicals, respectively. A broad array of organic compounds, both natural and human-made, can be metabolized by organotrophs. Reduced forms of numerous inorganic elements, including nitrogen, iron, manganese, sulfur, selenium, and others, provide energy for a diverse group of lithotrophic bacteria. On the basis of whether they obtain their carbon from carbon dioxide or organic chemicals, organisms are classified as autotrophs or heterotrophs, respectively. Organisms that respire are categorized as aerobic if they use oxygen as an electron acceptor, and anaerobic if they use other compounds as electron acceptors. These alternate electron acceptors include oxidized forms of nitrogen, iron, manganese, sulfur, selenium, and carbon dioxide. The terms strict and facultative are used to designate whether an organism is exclusively aerobic or anaerobic or able to live under both conditions, respectively. Viruses, though not technically classified as forms of life, are often considered in soil microbiology. They range in diameter from 0.02 to 0.25 microns. Although viruses are obligate parasites of the bacteria, fungi, plants, and fauna, species that inhabit the soil, many can persist outside their host, in some cases for years. Viruses may play a role in regulating microbial populations, though parasitism, and are believed to be important vectors for the exchange of genetic material among prokaryotes. Bacteria are a very diverse group of organisms in soil, and most major taxonomic groups are represented in most soils. As many as 13,000 bacterial species are estimated to be in soils based on analysis of DNA. Actinomycetes are most abundant at higher or neutral pH than under acidic conditions. Previously specific groups of bacteria that were thought to be most abundant in soil corresponded only to those organisms that could be cultured on laboratory media. Gram-negative bacteria, spore-forming bacteria, and actinomycetes. More recent methods that describe microbial communities based on extraction and characterization of DNA, e.g. cloning libraries, contradict some of these previous assumptions about soil organisms and indicate that some of the most abundant species have never been described. Bacteria range in size from approximately 0.3-1.0 microns in diameter and exhibit a variety of morphologies, including rods, cocci, and filamentous forms. Bacteria are usually smaller when living in soil, where nutrient conditions are poor, than when grown in nutrient-rich laboratory media. Although many soil bacteria are potentially motile, their movement is constrained by the low moisture conditions usually present in soil. Numbers of bacteria usually range from 10 to the power of 8 to 10 to the power of 9 cells in a gram of soil from the surface horizon, and the total biomass of bacteria usually ranges from 400 to 5,000 kg in a hectare of agricultural soil. Many species form spores or other types of resistant bodies that permit them to endure harsh conditions in soil. Soil fungi are a diverse group of a broad range of morphologies and life cycles in many ecosystems, such as forests. They constitute the largest biomass, 500 to 5,000 kg/HA, of all the soil organisms. In fact, the largest individual organism known, inhabiting a total of 2.5 square miles, is a soil fungus inhabiting a forest soil. Fungi are far less diverse metabolically than are the bacteria. Most fungi are organoheterotrophs. The most common fungal growth form in soil is mycelial growth. The mycelium is defined as a filamentous network of hyphae that grows by apical extension. Filamentous organisms, particularly fungi, are uniquely suited to colonize large volumes of soil containing heterogeneously distributed food sources. Some fungi can endure harsh environmental conditions or predation through having resistant structures such as spores or because their hyphae are made up of recalcitrant organic molecules. Reproduction can be through dissemination of sexual and asexual spores. Many plant pathogens are fungi, many of which spend at least part of their life cycle in soil outside a plant. Beneficial in some cases, necessary symbiotic relationships occur between mycorrhiza fungi and most plant species. Fungi are usually more abundant than bacteria in soils that are not physically disturbed, that receive large inputs of complex organic matter, and where organic materials remain on the soil surface. Because these phototrophs are sensitive to desiccation and because sunlight cannot penetrate far into the soil matrix, algal and cyanobacterial population densities are usually low except in flooded or poorly drained soils. Algae and cyanobacteria may be the major primary producers in ecosystems where plant growth is limited by low temperature or moisture. In deserts, for example, blooms following rainfall events provide the main sources of carbon and nitrogen to these systems. The soil habitat exhibits substantial heterogeneity, which in turn fosters and maintains an enormous biological diversity. The spatial distribution of microorganisms is far from uniform, as evidenced by the fact that microbial populations occupy only a few percent of the total mass or volume of soil. The heterogeneity in the distribution of microbial populations is manifest at the scale of the soil particle and aggregate within the soil profile and at the field scale. At the scale of micrometers to millimeters, soil consists of mineral particles complex with organic material and of the voids, pores created by their arrangements in space. The organic matter fraction soil ranges from less than 0.1-80% on a volume basis, with most surface soils having organic matter contents between 1 and 7%. Microbial biomass usually makes up 3-5% of the total organic matter in soil. The mineral particles, clay, silt, and sand, are components of larger structures called aggregates in which the particles are held together by microbial polysaccharides and hyphae. Soil texture is defined by the relative proportions of clay, silt, and sand. Soil pores between and within aggregates containing either gas or solution, often in an approximately equal ratio. The pore size distribution depends on the relative proportions of clay, silt, and sand, size particles, and it strongly influences which type of soil organisms are active. Numerous microenvironments differing in redox status, pH, nutrient levels, pore size, and other factors can coexist within microns of one another. Microbial distribution at the microscale is a function of soil pore size and access to the organisms to nutrients and electron acceptors. The size of a microorganism strongly influences location. Bacterial cells range from 0.3 to 1 micron in diameter, and 1-2 micron long for non-filamentous forms, and up to 15 microns long for actinomycetes. The diameters are similar in size to large clay particles, and are small enough to permit bacteria to inhabit the small pores found within soil aggregates. Fungal hyphae range in diameter from 2 to 10 microns and their lengths vary considerably. Being too large to penetrate into most soil aggregates, fungal hyphae grow on the surface of and between aggregates. This is due to the fact that nutrients and oxygen are consumed by organisms living at the surface of the aggregate. Organisms capable of anaerobic respiration, such as denitrifiers, are often active within the anoxic centers of soil aggregates. Most methods for measuring soil properties are for relatively large volumes of soil and lumped together phenomena occurring in numerous microenvironments. Highly sensitive techniques capable of making measurements at spatial scales relevant to microorganisms are usually not possible to employ in complex systems such as soil. At the scale of millimeters to meters, there is a horizontal pattern in the soil's composition with a higher organic matter content in the surface which declines with depth, and higher amounts of clay deeper in the soil profile. Generally, the density of microbial populations declines with soil depth, primarily because of corresponding decrease in organic carbon content and to a lesser degree in oxygen concentration. There is also a lateral and horizontal pattern in the composition of soil that is created by the spatial distribution of plants. Plants, particularly their roots, are essential members of soil communities and an important source of carbon and other nutrients to microorganisms. The rhizosphere is defined as a zone of soil under the influence of plant roots. The soil not contained within the rhizosphere is commonly referred to as bulk soil. The rhizoplane is defined as substances and soil particles and is found at the interface between plant root and soil. Rhizosphere communities contain larger population densities, greater microbial biomass, and higher levels of certain types of microbial activities than do adjacent communities in the bulk soil. Gram-negative bacteria and denitrifiers are cultured in greater numbers and rhizosphere than bulk soil samples. Fungi, particularly mycorrhiza and plant pathogenic species, are also abundant in the rhizosphere. Conflicting data prevent making conclusions about whether microbial communities are more diverse in the rhizosphere or bulk soil. As tools for characterizing microbial communities continue to improve, we will have the ability to answer such questions. Good day, this is Professor Will, CBMD PhD. Today is Thursday, November 21st, 2019. It is 6:23:25 Eastern Standard Time. And this concludes my reading on microorganisms. Hope you enjoyed listening. Have a great day.